So I'm really happy to introduce Dr. Avery Berman. He earned his MSc in medical radiation physics and PhD in biomedical engineering at McGill University, both under the supervision of Professor Bruce Pike. His PhD thesis was on the development of advanced biophysical models to extract quantitative physiological parameters from the bold functional MRI signal. In other words, he investigated the physics of how blood gives rise to MRI detectable signals. Though we've had a general picture of the biophysical underpinnings of functional MRI for some time, the details remain active areas of research, and the specifics in terms of fMRI's ultimate spatial and temporal resolution remain unknown. Avery's current work with Dr. John Pulmeni at the Martino Center is changing how the field thinks about fMRI at high resolution, and we'll hear about this exciting work today. On a personal note, Avery is the sort of guy who you really want on your team, whether it's in the lab, on the pitch, or navigating life in this wild world. I've really been looking forward to your talk, so take it away.
Wow, Jeff, that's a great introduction. Thanks so much. Let me make sure I can share my screen too here. Okay, everyone sees that?
Yep, that's good.
Okay, great. Yeah, it's really a pleasure to be here. I wish we could be in person, but such is life. And I'm also very close to the children, so when we're able to, I'd love to come and visit. Uh-oh, is there timing on? Anyways, what I was going to say was that I don't have any personal experience with pediatric medical imaging. However, I think I can relate to some of the issues that you may encounter during your studies. And so the outline for this talk, as Jeff has mentioned as well, I'm going to dive into sort of some of the nuts and bolts of the acquisition that we use for high resolution fMRI. More specifically for bold fMRI. But first I want to back up and also motivate why it is we think we should be doing high resolution fMRI in the first place. Then I'll quickly touch on some non-bold applications that we've been using the new pulse sequence, which I'll describe, called VFA-FLEET. And then finally, I want to actually go over sort of this merging of understanding the role that the image encoding that we typically use, mainly echo-planar imaging, how it impacts the contrast that we're trying to attain, such as bold. Gradient echo bold is what most people are familiar with, but spin echo bold has also long been touted as a sort of holy grail of fMRI. And so I'll kind of dive into why we think it's something interesting to look at and how image encoding impacts then our contrast encoding. So first, some of the background material. So for decades, fMRI has been non-invasively used to map the macroscopic organization of the human brain. So we've generally used relatively coarse voxel sizes of 2 to 3 millimeters, isotropic, and then we can map out like this large scale organization of the brain. But there's growing demand, as well as emerging techniques now with MRI, to understand the functional circuitry of the human brain at the so-called mesoscopic scale, where neurons are anatomically or functionally or sometimes both structured at spatial scales that are much smaller than what we typically can image with fMRI. So we're interested in looking at cortical layers and cortical columns, as well as nuclei of the central nervous system, which are typically sub-millimeter scale. But there's often been this idea or this notion that bold imaging has a very coarse biological resolution, and that we can only actually resolve changes going on in the vasculature, which is what we're using to indirectly infer neural activity. And we can only resolve changes at the spatial scale of 2 to 4 millimeters. But there's now growing evidence from optical techniques mostly that actually the biological resolution of the hemodynamic response of neurovascular coupling is much more spatially and temporally precise, as Jeff was alluding to initially. And so I want to give some of the kind of current optical imaging results that are actually informing our understanding of the spatial resolution of bold fMRI. So some of the earliest or most convincing work has come out of Anna DeVore's lab, as well as work from David Kleinfeld at UCSD. using two-photon microscopy. And they were looking at diameter changes of individual cortical arterioles as they did a 4-paw stimulation to rats and mice, actually, in these 2 different studies that they performed. And what they found was that the deepest arteries near the white matter boundary would often be the first, most prompt to respond to this stimulus, as shown by this red curve here. Whereas, and then this dilation would propagate upwards towards the surface arterioles, which are generally larger and further from the source of neuronal activity. So this initial prompt response from the deeper arterioles is actually much more neuronally and spatially specific. But of course, with fMRI, we're actually not imaging arteries. We're more sensitive to deoxyhemoglobin in capillaries and veins. And so they were actually able to confirm their results in a gradient echo bold fMRI study where they looked at doing the same experiment, and they looked at the change in bold time course across depths of the rodent cortex. And they found a very similar trend as shown in this plot on the left of the onset time for this bold response, where they found that the deepest layers had the shortest time to onset, and then this propagated upwards towards the upper layers. So even on the venous side of things, we're also seeing this sort of spatio-temporal cascade of events, which is really encouraging if we're wanting to resolve changes in neuronal activity within the cortex, and not just from these kind of large vessels that lie across the peel surface. So yes, to summarize, the mid to deep bold response onset is first, as shown here, and then the surface bold response is last. And now there's actually even more recent work coming out of Mike and Nedergaard's lab that's suggesting it's not just the vessels themselves that are responding to changes in neural activity, but also the blood or the red blood cells also have a response. And so this is some newer results that I came across in the last year or two that really blew me away, and I'll try to explain them here. So they were measuring the time of red blood cell velocity change in both capillaries and arterioles when they did a hind limb stimulation in this case. And what they found is that when they look at the onset time for the RBC velocity to change, the capillaries onset time was nearly 2 seconds shorter than that of the arterioles. So the arterial onset time is, and then if I also look over at the diameter change then of these vessels, they found that the arterioles were dilating at approximately 2 seconds after they stimulated the hind limb. So what we see then is that the capillary velocity change is preceding the dilation of the arterioles as well as the increase in red blood cell velocity by approximately 2 seconds. So these capillaries are sensing changes in neuronal activity much more rapidly than what we see through the vasculature itself. And the mechanism that they proposed for this, when they actually look at this ex vivo then, is if they change the partial pressure of oxygen in the medium that the red blood cells are flowing through, the red blood cell velocity would increase as the PO2 would decrease down to 0 oxygen in the solution. And so the mechanism that they proposed for this was that they also measured the change in the elongation of the red blood cells. So the diameter of the red blood cell to the thickness of the red blood cell. And as they decreased the partial pressure of oxygen, they found that these red blood cells would elongate, which allowed them to flow much more rapidly through the tight capillaries, therefore increasing their flux to deliver more oxygen to the regions where there's increased brain activity. So to summarize that, the RBC deformability increases under low partial pressure oxygen conditions, which can therefore in turn increase the velocity of these red blood cells so that they can promptly meet this demand for more oxygen. So I tried to, just for my own sake, try to make a chart of what is this kind of cascade of events in the hemodynamic response. And so the way I see it is that we have this initial increase in neuronal activity, which then drives an increase in metabolism, both for oxygen and glucose, which in turn decreases the partial pressure of oxygen of tissue, which as a result then increases the red blood cell velocity, which through a passive mechanism. At the same time, the increased neuronal activity is sending signaling molecules and neurotransmitters that then results in this precapillary arterial dilation. But this is coming, the onset of it is actually after this change in the red blood cell velocity, which then this propagates upstream through the arterioles. And then finally, we get kind of the classic downstream venous dilation. And so for me, adding this RBC velocity thing was actually a new ingredient in my understanding of the bold physiology. And so to summarize this, we say that the hemodynamic response spreads in time. There's mechanisms for endothelial cells of the vasculature to spread this signaling up the pathway to provide kind of just-in-time changes in blood flow to regions that require it. And the early response may be more neuronally specific. Let's see. Yeah, and this early response is really seen in the red blood cell velocity as well as precapillary arterioles. But we'd say that we still don't know what the biological limits for fMRI are, both spatially and temporally. So this 2 millimeter full width half maximum bold point spread function, I think is no longer up to date, and we still haven't driven down to what is the resolution we can resolve brain activity with bold. But in addition to these physiological considerations for why we want to move to higher resolution, I think there's also reasons in terms of our data analysis as well as our interpretation of the bold signal that motivate us to move to higher spatial resolution. So I'm showing here an image that I acquired with a bold fMRI image acquired at 0.8 millimeter isotropic voxel size, and I've downsampled it by a factor of 3 to a more conventional voxel size of 2.4 millimeters isotropic. If we just zoom in on one area of the brain, what we can see is that there's all of these veins here within the sulcus of the cortex, and we don't see any of them whatsoever on this downsampled version. And since we know that the bold fMRI signal is coming largely from veins, entirely from veins really, and capillaries, this would have a very large influence on just our interpretation of the bold signal and where exactly it's coming from. In addition to that, we also can get several samples of voxels across the cortical depth here. And so we can Nyquist sample the cortex, which allows us to then to tease apart different physiological effects if we want to get just gray matter signals and not include signals from CSF or from white matter, which will then not potentially be showing the bold signal of interest. Okay, so hopefully this helps motivate why we want to move to high resolution. Now I'll discuss some of the developments we've made in order to move to these higher spatial resolutions. So first off, if we want to image at higher spatial resolution using a typical echo-planar acquisition, and we wanted to double our in-plane spatial resolution, for example, this means that we'd have to cover 4 times the area in a given readout, which is then, as a result, going to take almost 4 times as long to acquire. And we know that when we have long readout durations, this results in increased geometric distortion, spatial blurring, and also gives us a suboptimal contrast to noise ratio because it tends to push out our echo time for bold imaging. We can use parallel imaging to try to cut down on this readout duration. However, to get to these sub-millimeter spatial resolutions, we still need high undersampling factors that result in too many artifacts in our images and low SNR. Another question we could ask is, can we just scan faster? Can we just ramp our gradients and switch them on and off faster to get through cases more quickly? And the short answer is yes, we can. However, a current limitation is the human body. If we ramp these gradients too quickly on typical whole body scanner systems, we get peripheral nerve stimulation, and this is a major concern for patient safety and comfort. And so we actually can't scan faster as a result. So we say that today we're encoding limited in that in order to get to these sub-millimeter voxel sizes, we just don't have the ability to image our volume quickly enough without imposing too many artifacts in the images themselves. So one technique for overcoming this readout duration limitation is to go back to multi-shot echo-planar imaging, which used to be a popular technique before parallel imaging came into vogue. And so what this does is it segments our EPI readout across multiple shots, in turn reducing the readout duration of any given shot. But we know that the order in which we loop over slices, if we're doing a multi-slice EPI acquisition versus segments, has a dramatic impact on image quality. So if we're to loop over slices first, which is kind of the conventional segmentation technique, there's typically a long delay between acquiring segments of any given slice on the order of 1 to 2 seconds, leaving our acquisition then vulnerable to the subject to move in that time frame, or even just have changes in our B0 field due to respiration. And this results in intermittent ghosting artifacts, which degrades the temporal stability of our fMRI data set, making it relatively unusable. Alternatively, we can loop over the segments first, and this is a technique called FLEET, or fast low angle excitation echo-planar technique, proposed and developed by Sir Peter Mansfield's lab in the 80s, actually. So we're going back to an old technique as a result. And this reduces that intermittent ghosting because we acquire segments for any given slice sequentially. And this has been used for FLEET ACS acquisition for graphic training and reducing the resulting artifacts in the training data and improving the TSNR of our fMRI data. But this FLEET ACS data typically has lower SNR. And so if we want to maximize our SNR, we actually have to use a variable flip angle scheme, which has had some challenges in the past, which I'll explain here. So if we want to use a variable flip angle scheme, we want to maximize our SNR, then we'll want to finish all of our segments on a 90-degree excitation pulse, so that we use up all of our longitudinal magnetization. And so just to show you what happens if we apply this variable flip angle scheme, we would initially start with a smaller flip angle. And if we just use our vendor's sync pulses, and we apply that to our slice, then we'll get a certain slice profile. This looks very normal. And then we will scale this sync pulse to achieve a slightly elevated flip angle. And if we apply it, then we get a similar looking slice profile, but it's now broadened. And then finally, if we end on this 90-degree flip angle, which is going to use up all of our longitudinal magnetization, giving us maximum SNR, and we apply it, then we end up with this very broadened and distorted looking slice profile. And the only place we actually get the desired signal intensity is right at the center of the slice. But since we're taking signal from across the whole slice, it means we're going to have changing signal intensity across segments, which is going to result in ghosting. So we've developed a recursive RF pulse design based on the Chenard-Leroux algorithm, in which now if we apply our first RF pulse, we get some slice profile. If we apply our new RF pulse, we end up getting the same slice profile. And then finally, if we apply our 90-degree RF pulse, we get the same slice profile. And hopefully you can see from this that these are no longer just scaled versions of each other. There were initially negative lobes on either side of this peak for the 35-degree pulse, and we no longer even have that for this 90-degree pulse. And just for any of the RF experts in the audience, I wasn't sure exactly who would be here. I just wanted to quickly go through exactly how this is done. So this was in collaboration with Will Grissom, who's an RF pulse design expert from Vanderbilt. As I'd mentioned, it uses the SLR algorithm. And so what we do is we provide our target profile parameters, which is like time bandwidth product or flip angle for the pulse. And we provide that to the SLR algorithm. And so we take the inverse SLR transform, which gives us our first RF pulse. And then we apply our forward SLR algorithm, which then gives us our slice profile in MXY, as well as longitudinally along MZ. And this was the real critical ingredient, was getting how much longitudinal magnetization remained after we apply our first pulse, because we then pipe that back into our algorithm, and we can again apply the inverse SLR algorithm on that. And as a result, then we can see how MZ is changing across every shot. And tailor our B1 profile, or our B1 waveform, so as to get consistent transverse magnetization profiles. So now let's take a look at some of the data. So I'm going to show here some 1 millimeter isotropic fMRI data acquired at 7T using either, and it was using 2 shots, as well as further in-plane acceleration of a factor of 4. And so this is using the conventionally segmented acquisition where we loop across slices first. And what we can see here then is a lot of flickering in the image that's intermittent. So it means it's hard to predict necessarily when it will happen. It's motion related, and it's also not motion related, and it can be just induced by B0 changes from respiration, which are amplified at 7T. If we then perform this VFA-FLEET technique, the variable flip angle FLEET technique, using our scaled vendor pulses, the scaled sync pulse, we no longer see this intermittent ghosting. However, we still see some stable ghosting that's resulting in this general non-uniformity in image intensity throughout the slice. And then finally, if we do the VFA-FLEET acquisition using our recursively designed SLR pulses, we've reduced both the intermittent flickering that we see, intermittent ghosting that is, as well as the stable ghosting, and we have much better image uniformity across the slice as a result. And so now we can actually quantify this change in the images by looking at TSNR maps. And we can see just in the TSNR map of the conventional segmented, as well as the VFA-FLEET using our scaled sync pulses, large non-uniformities in the TSNR that are not necessarily anatomically driven or driven by just our coil sensitivity receiver profiles. And so as a result, this makes it harder to interpret fMRI results if we're looking at these acquisitions after the fact. If we don't see activation in a certain region, is that because that region was not activated, or because we just didn't have sensitivity to detect the activation anyways? And most of these non-uniformities are gone in our recursive SLR pulse design. So we're really encouraged by these early results. It actually surprised just how well they worked as we implemented them. So then we wanted to really push the envelope here and go for kind of the highest spatial resolution that we could think of that might be feasible. And in this case, it was a 0.6 millimeter isotropic acquisition. We then performed a flickering checkerboard type of experiment of just 30 seconds off and on alternating pattern. And we achieved this using 3 shots and an in-plane acceleration factor of R equals 4, meaning that any given shot of our EPI acquisition is undersampled by a factor of 12. Our volume TR was relatively long for this number of slices. It was still just under 6 seconds. And also want to emphasize that we took kind of no shortcuts in either the acquisition or the analysis here. So we did not perform partial Fourier so as to get echo time down. Oh, and sorry, I didn't put echo time, but it was 27 milliseconds. We didn't use zoomed imaging, which is often used to reduce our field of view and then reduce the number of phase encoding steps. And then the analysis, we performed no spatial smoothing. And so if we take a look at the results here, we can see robust activation across 3 different subjects, all with 3 very different slice prescriptions. It's generally confined to the gray matter as well as to the cortical surface, which is a known bias of gradient echo bold. And also want to point out that these underlays here are the EPI images themselves. So this is the average across a single run. We performed 3 runs per subject. And so these results are very encouraging as well, but of course we don't actually just want to look at flickering checkerboards in our studies. We're much more motivated, like I said, to look at more of the laminar and columnar organization of the brain as well. So some of the take-home messages for this part here is that using this variable flip angle FLEET with an RF with a recursive RF pulse design, this has enabled us to perform ultra-high spatial resolution at ultra-high field down to a voxel size of 0.6 millimeters isotropic. This had reduced image artifacts from spatial blur and distortion, as well as intermittent ghosting that we see in conventional EPI, segmented EPI, as well as these slice profile consistencies that we see when we don't use the recursive pulse design. One of the limitations of this technique, though, is, of course, the temporal resolution, because our volume TR is now extended by however many shots we use. So I've implemented a SMS version of this as well to help reduce the repetition time and or increase the amount of brain coverage that we have. And so now I also wanted to touch on a few of the non-bold applications that we've been now looking into using this sequence or similar sequences as well for. So we've been using this exact sequence now to do cerebral blood volume fMRI using contrast agent ferromoxetol. And so for those unfamiliar with ferromoxetol, it's an ultra-small paramagnetic iron oxide or USPIO contrast agent that shortens both T1 and T2 star. And the amount of shortening is directly proportional to the blood volume. It's also has a long circulating half-life in the blood plasma on the order of 14 hours. So as a result, we can actually do continuous CBV-weighted fMRI as opposed to, say, a dynamic susceptibility contrast style experiment with GAD. And doing this, we get improved sensitivity as well as physiological specificity over the bold acquisition. But the main challenge is that in order to minimize the bold contamination, we also need to minimize the echo time. And this has been challenging for high resolution studies in general. So we used our VFA-FLEET sequence, and in collaboration with Jingwan Chen, who's also a postdoc in our lab, we were able to perform some simple flickering checkerboard experiments, and we get robust activation that's even more confined to the gray matter, I'd say, than my bold acquisition that I showed before. And so we were able to acquire data at 0.8 millimeter isotropic, again, using 3 shots in plane acceleration of 4, which helped us get our echo time down to 17 milliseconds as a result to try to reduce some of that bold contamination. And again, this is showing the mean EPI image over a single run. And then the overlay is showing the mean activation over 4 runs. And we know that this is a CBV weighted acquisition because our bold signal changes are now negative as opposed to the bold effect, which is positive. Because as CBV increases, we get more T2 star dephasing, and so our signal goes down with the stimulus here. Another application that we've been using is non-fMRI related. It's actually to perform distortion-free diffusion-weighted imaging as well as rapid anatomical imaging. This is in collaboration with Jae-Jin Cho and Birkin Bilgic, as well as Boryan. And we've proposed this technique called voodoo, which is variable flip angle fleet ordered, blip up, blip down, undersampling, in which we do 2-shot EPI acquisition, where the first shot we acquire with a blip up EPI case space acquisition. And then the second shot, which is the 90-degree pulse, we do blip down. And so as a result, we see 2 different distortions in the images if we can reconstruct them separately. And then if we apply top up to these, we can get a field map, and we can plug that field map into a forward reconstruction model and also perform a Hankel low rank constraint so as to impose some consistency across the 2 shots as images, and then reconstruct a nominally distortion-free EPI image. And so we've applied this now to spin echo EPI for T2 weighted anatomical imaging, where we can see that there's differential distortion in the blip up versus blip down shots at these 2 different flip angles. That's mostly resolved in the voodoo reconstruction in this slice here on the top, as well as the slice on the bottom. These arrows are pointing out, though, that there are different image intensities across the 2 different shots as images. And one of the main reasons for this is that we did not use our recursive pulse design in this. So that would be a next step for us, is to actually incorporate the recursive pulse design. In addition to just T2 imaging, we've also applied this now to diffusion-weighted imaging. And so this is just showing 6 different directions at B equals 1000 value. And so importantly, one of the important factors here is that the time between acquiring the segments was just 113 milliseconds. So this makes us much less vulnerable to motion that might occur between acquiring these 2 shots. So just to quickly summarize, I've shown that the VFA-FLEET acquisition is compatible with a range of contrast preparations, from T2 star for classic bold, but also to T2 diffusion-weighted imaging, but even also T1 weighted imaging if we do some sort of contrast prep like an inversion pulse, similarly for ASL. And this will further benefit when we apply our recursive RF pulse design into the system, into the sequence. Okay, so now I've discussed a fair bit on the pulse sequence and the acquisition side of things. I also wanted to spend a bit of time now to study this interplay of the image encoding, so kind of the pulse sequence side, but also then from the contrast encoding that we perform, which is more from the biophysical and physiology side of things as well, with a particular emphasis on spin echo bold. So it's well known that spin echo bold, as shown in the early 90s by Boxerman and Bruce Rosen in the group at the Martino Center, well, the NMR Center at the time. So it's well known that spin echo bold has lower macrovascular sensitivity than gradient echo bold. And so it has peak sensitivity to microvessels or capillaries, which then imparts it with improved neuronal specificity as a result, since the capillaries more closely, in more close contact with the neurons themselves. However, if we just acquire a spin echo bold acquisition and we look at the cortical depth dependence here of activation to a flickering checkerboard pattern, we see that there's this expected peak in the midway through the cortex of this. This is a monkey cortex study as well. But what we also see is actually a very large and prominent level of peel contamination, which would be unexpected if we're getting this pure T2 acquisition that we desired. And the way this data was acquired, it was using 4 shots of a segmented spin echo acquisition. This was prior to animal scanners really having kind of the hardware and the acquisition to perform accelerated parallel imaging. So they were actually doing segmented imaging at the time. And so they acquired with 4 shots, making their readout window around the spin echo to be 30.7 milliseconds per shot. And what they found is that they increased to 16 shots and reduced that readout window to 7.7 milliseconds. They were able to get rid of almost entirely this peak at the peel layer. So they're able to get rid of that macrovascular contamination. And the source of this macrovascular contamination is that there's now R2 prime weighting. The further we read out away from the spin echo, there's R2 prime weighting at the edges of K-space, which is encoding that high spatial resolution information. And so we didn't initially predict this with our bold biophysical simulations because we typically only looked at signal at the echo time. So a goal of the study here, which I'm presenting this year at ISMREM, was for us to better understand the role that the EPI acquisition window has on bold simulations, as well as in practice, how we can then try to optimize our acquisitions. So to achieve this, we added temporally varying phase encoding gradients into biophysical simulations of the bold signal from a vessel network, as shown in the top here. And this vessel network was then embedded into an image slice, you could say, where there's no magnetization surrounding this voxel. And we're just trying to look at how the signal varies from this voxel at the center, which is the vessel network, as we change things like the echo spacing between these phase encoding blips, which then will change the total readout window duration. We can then reconstruct an image or a profile of the image across the image slice, which just uses basic K-space formulation between image domain and K-space domain, but in turn just turns out to be an average of our signal at every echo in this phase encoding in the EPI echo train. And we also looked if we could just retrospectively add in this effect of the phase encoding gradients rather than physically adding in these gradients into the simulation. And so we wanted to do 2 separate sorts of experiments. And the first was to just recreate those Boxerman plots that we see of this peak microvascular specificity as a function of vessel size. And so we did this, we calculated percent bold signal change versus vessel radius. But in addition to that, we added changing our EPI readout window length. The second experiment we wanted to do was to recreate this Gunza and Logothetis study where they showed the decreasing macrovascular contamination as they reduced their EPI readout window. And so we did a depth-dependent sort of peel vein contamination study, again with changing readout length. And so just looking at some of the results from that first aspect of the study where we're trying to reproduce Boxerman, I'm showing here on the left our percent bold signal change from the spin echo acquisition as we increase vessel size. And so this is kind of like the ideal case where we could sample just at the spin echo. This black curve is also showing an even more ideal case of what the signal would look like from a different set of simulations not shown. And we see that all of our simulations tend to fall along this ideal line when we image right at the spin echo time. But as we start to increase our EPI readout duration, we start to introduce more and more large vessel contamination. Although it's still not so much, it's still far below, the amount of contamination we're getting is still far below the actual peak from the microvessels. And so if we increase our readout duration even longer, we see again an increase in this contamination, but also more importantly is an increase in the variance of the contamination. And so this has really important implications for if we have a single large vessel in a voxel, it can impose a large kind of partial volume contamination then of our desired bold signal. And so next, in order to model this cortical depth dependence of macrovascular contamination, trying to reproduce that Gunsa and Logothetis study, what I did was we simulated the microvascular signal from a voxel that contained just 4 micron radius vessels. And we moved it relative to a peel vein that was lying just outside of it, not inside it actually. And we moved it and we saw, and we simulated separately what was the peel vein, the signal coming from just the peel vein, as well as the signal coming just from the microvasculature. And we even applied these physical gradients in 2 different orientations as well, both tangentially and radially. The reason for this is because the peel vein was aligned so that its B0 field would have a maximal effect on the microvascular network. And so we wanted to see then how then the phase encoding gradients might interact with this intrinsic field offset from the peel vessel itself. And then finally, we wanted to determine the depth at which the peel vein and the microvascular bold signals were equal. So at what depth did the microvascular start to take over from the peel vein? And so here we're seeing these results where, again, this top plot, if we just focus in on it, this is showing the bold signal change from the peel vein, which is the colored curve here, as well as from the microvasculature. And so when we're very far from the peel surface, we get just a microvascular bold signal change on the order of 2% here. We get no peel vessel bold signal change. But as we move towards the surface, we start to introduce this peel vein bold contamination. And at the same time, we actually see a decrease in the microvascular bold signal. And this is because we start to get partial voluming with voxels or with regions above the peel vein, above the peel surface, where there is no microvasculature. So essentially just CVV is decreasing here. And so I also want to point out that there is even at this ideal case where we're measuring just at the spin echo, we still get a peel vessel bold contamination up to some depth within the cortex. So we still aren't getting just signal from the microvasculature. But even worse than this is if we increase our readout duration to say 32 milliseconds, the peel vessel contamination shoots up to nearly 40% bold signal change, which isn't really realistic or seen in practice, likely due to our CNR would just get so low we wouldn't actually be able to measure signal change with that. But it takes much longer then as a result for that peel vein contamination to come down and to go below this threshold of the microvasculature. And so if we quantify where it is that these curves cross, we can see where the microvascular dominance really starts to set in. And so this is just showing what is that critical depth in which the microvasculature starts to have a larger bold signal change relative to the peel vein. And so for 0 milliseconds, they cross over in close to 200 microns depth into the cortex. But if we go up to 32 milliseconds, which is quite a conventional type of EPI acquisition, we have to go nearly 600 microns into the cortex before the microvasculature starts to dominate the peel vein. And if we go even longer, it just gets deeper and deeper as well. So using this sort of information, we can start to get a quantitative understanding of the role that the image encoding has on our contrast encoding and try to devise then kind of an ideal acquisition that's a compromise between getting the contrast we want, but in a realistic amount of time or realistic amount of spatial coverage. So to conclude this part, we've proposed this new framework. So this is the first application or implementation, to my understanding, where we've actually included both biophysical simulations with image encoding gradients to provide increased accuracy in the simulation of the bold signal. As also, we want to use this to help guide acquisition strategies. Also, this is the first time I've seen someone take these infinite cylinder models to do bold simulations and just add in more imposed realistic structure as well by placing this peel vessel outside of our voxel. It's very simple, but because of the simplicity as well as the accuracy of these infinite cylinder models, it does help us with the interpretation of the results as well. But infinite cylinders are great for these sorts of interpretation reasons, but they're not really realistic. So we also plan to incorporate this into 3D vascular anatomical networks, like what David Boas's group has been developing for a few years now. And there's several other applications in where we don't have these this great data from monkey cortex of spin echo. So there's several other pulse sequences that have been developed where we don't really have a good sense of how the image encoding is impacting the contrast that we're trying to achieve. So sequences that we're interested in applying this to include 3D Grace, center out EPI, as well as spiral EPI, because all of these have some sort of T2 star contamination in whether they're trying to image T2 bold effects or ASL at VASO. And so finally, also to conclude now, I hope I've shown that we're now in the middle of a multi-shot EPI renaissance for both fMRI as well as anatomical imaging. Using this FLEET technique, we can have reduced motion sensitivity that really plagued conventional segmentation for a long time. And we can also, using the variable flip angle scheme, have high SNR as a result. This has allowed us to go to much higher spatial resolutions than previously possible. And if we combine these effects, then with the spin echo bold acquisition, we can also achieve higher spatial specificity to capillaries and, as a result, neuronal activity. We can also combine this acquisition, as I've shown, with advanced reconstruction techniques to perform rapid anatomical scanning, like T2-weighted or diffusion MRI, with both reduced distortion, as well as motion sensitivity. So this is a very versatile pulse sequence. We can generate quite a wide range of contrast types. So for future applications, I think it's hard now to discuss a high resolution fMRI without discussing 3D echo-planar imaging, which is really actually almost taken over in some cases for 2D EPI. But these variable flip angle recursive pulse design techniques that I've presented here are fully compatible with 3D EPI as well, actually, and could help give us better slab profiles as a result for sequences like VASO and ASL. And the profiles could then be more stable across segments or partitions, and also help maximize then SNR. Also, I didn't mention any of the B1 plus inhomogeneity issues that we ran into with the VFA-FLEET, but there were several. And actually, we had to leave a lot of SNR on the table in order to get these relatively decent looking acquisitions. So we plan to implement a parallel transmit version of these sequences or these pulses as well, to start getting back this SNR that is really a main motivation for going to ultra high fields. And so, as I mentioned at the beginning, I don't think that we've reached the biological resolution of fMRI quite yet with our imaging resolution. And actually 0.6 millimeter isotropic is still not enough. We have plans to go to higher spatial resolutions on a head-only gradient system that's being designed and developed by David Feinberg at UC Berkeley. And there we're hoping to get to at least a voxel size of 0.5 millimeters. So we're really excited. And this will also tie into some of the developments for parallel transmit as well. So we're really excited to start looking into these even higher spatial resolutions. But as we push to higher spatial resolutions, we're still going to need clever encoding strategies to reduce this contrast contamination coming from these long EPI or whatever readout method that we choose to use. And so with that, I'd like to thank you all for the invitation to present to your group. I'd like to thank my collaborators, my funding sources, and I'm happy to take any questions.
Thank you so much, Avery. Ellen, did you have any questions? I know you need to pop off here shortly.
Oh, it's just the first part was really interesting. I'm sure you just thinking about what happens in our sickle cell kids too, and how interesting it would be to get them at high field and understand velocity changes, and whether we see the same kinds of things when their red blood cells can't deform as much. I think it would be really interesting. I'm curious, in terms of all of these things that you've learned at high field, do any of these trickle down to lessons that can be learned at 3T as well too, in terms of optimizing acquisitions?
Yeah, I've thought about this a fair bit, especially in the scope of the faculty hunt as well, where 7Ts are still rare. And so I think there are actually a lot of 3T applications of these pulse sequences as well. So all of the anatomical imaging that we did, that diffusion-weighted imaging, the T2, that was all at 3T as well. And actually these pulse sequences in a way will work better out of the box at 3T, because we don't have these B1 plus non-uniformity issues that are really challenging for this acquisition. So one pulse sequence, sorry, pediatrics out there. Sorry, one pulse sequence that's getting me really excited is ASL with reduced bold contamination. Because the main point of doing this ASL studies is we really want to get like a perfusion signal, but bold is always riding along when we do these EPI acquisitions. And so we can use different EPI trajectories that have much shorter echo times. And if we use this VFA-FLEET sequence, so it'd be a multi-shot acquisition, it would be a bit slower, but we can use VFA-FLEET to essentially get 0 or very few millisecond acquisition, get no bold effect, and see really just kind of like the T1 effect from ASL that we're really interested in. And groups have been looking into this as well. And we think that in combination with RF pulse design, they're going to get better image quality in addition to this reduced bold contamination. And it also improves just their sensitivity to the CBF change as you go to this lower echo times, and you get better SNR. So I think a lot of these non-bold acquisitions would be really beneficial for these types of sequences, like CBF and CBV imaging in particular.
Great, thank you. Wonderful talk. I really enjoyed it. And
thanks so much.
The other thing that would just caught me on a slide of other people ask questions is just and of course, from the neuroscience side, is really interesting that they feel the blood, see the blood flow changes at the deeper layers before the upper layers. Those are the ones that form first and have the connections to deep structures or projection fibers. So it's interesting that that is seen, and I just can't wait till we can start doing that kind of imaging on fetuses.
Oh my gosh. Are you saying with MRI?
Yeah, although I don't, practically speaking, we're stuck in but hopefully we'll come up with strategies to be able to look at blood flow, in a smaller head size as well too.
Mm-hmm.
Yeah, I was struck in your introduction, Avery, how the way you framed the problem was that, motion of the adult brain is actually why you're kind of driven to do this, more clever encoding scheme, with multi-shot ordered the way you do, which is actually the exact same thinking we use with fetal imaging, right? We have to go fast enough. That's at the end of the day, how we control for motion is by going fast. And it did make me wonder, I know that the encoding is fast enough, you can't fit in like prospective motion correction into that mix. But I'm there has been a lot of advance in prospective motion correction. Wondering if you see any way for those to kind of a confluence down the road here.
Yeah, I think we could fit prospective motion correction in. I guess with the FLEET technique, maybe it'd be more challenging because the excitations are. But I think I don't know what is kind of the most latest with prospective motion correction, but I think they only typically need just a few milliseconds to acquire maybe some extra data, whether if it's MR-based tracking or if it's optical, then it can be really quick, I think. So I don't think that that's necessarily a big lag for us. But also for us at 7T, it's not just motion, but just changes in B0 are really profound if we don't, if we acquire our segments too far apart in time. You can see that if you do a field map that's kind of gated to the respiratory cycle, you can see huge fluctuations, like, you know, tens of hertz fluctuations, maybe even more throughout the respiratory cycle that then impose this phase shift across shots, which then give us just kind of this flickering ghost pattern. The subject can be holding completely steady, not moving at all, and you still see this flickering. So it helps not just with motion, but other physiologically kind of driven changes that might be changing on a time scale of a second or so as well.
Does anyone else have any other questions?
I'm just curious, in when you're correcting for that respiratory motion, whether you can capture that as well too as another input or signal to track.
So people do that, I guess, currently with just general fMRI analysis. They use Gary Glover's method, retro I-core, and so they use external monitoring of respiration, like with respiratory bellows. And so you can get a sense then for what's kind of the periodicity of the respiration. And I know a group that was doing 3D EPI, which is quite different in a way. They were actually trying to incorporate the signals from the respiratory bellows into some sort of correction across partitions of their 3D EPI scan. I think we could, I'm sure also with machine learning now, like we could just find out, okay, what's the artifact coming from just this B0, like this gross macroscopic B0 change. I bet there are ways of teasing that apart as well. But I think trying to acquire good data up front is even, you know, and combining these other strategies is probably the way to really go. Yeah, I imagine that we could capture B0 from, or respiration at least, from both imaging as well as external monitoring, and combine that with, you know, good data and get even better data.
Owner, did you have a question?
Actually, I think he,
Maybe he'll type it.
Hello, can you
It's
hear
quite
me?
an echo. Yes.
okay, actually, I was gonna follow up on the physiological noise, right? Because although you have a very clever way of reducing the overall acquisition to reduce the time, right? You still have a multi-shot sequence, right? So you are still affected by physiological motion more than a single shot EPI, right?
Yes. Yep.
Okay, and then the other question I have is somewhat related. Do you do something specific to solve the phase inconsistencies between shots?
Yes. Okay. So I guess the first part of that regarding physiological noise, our excitation TRs are on the order of, say, 70 milliseconds. So if we're acquiring 2 shots, it's just taking 140 milliseconds to acquire all of the data.
Mm-hmm.
So the physiological noise on that scale is very minimal generally. So we still are much less sensitive to physiological noise as compared to something that takes a second to acquire all of the data. And then regarding the phase correction, I didn't go into this at all, but we do perform, so we perform this classic Nyquist ghost correction just for EPI readout positive lines versus negative lines. So that's a classic ghost in EPI. And so we perform that correction within any given segment. But then we also perform the exact same correction across segments. So if there's any phase difference across segments, we'll apply the same style of correction so as to remove any possible phase corrections. But the magnitude of those is also really quite small. More importantly, though, in not just the phase changes, there can be still some residual magnitude changes across shots. So we also, using these navigators, we'll try to reduce the difference between the navigators, find a scaling factor for that, and apply that to our imaging data. But that is not really a physiological effect. Sorry?
Do you acquire a navigator for each shot?
We do, yes. And that, I mean, maybe you're getting at this, but we could probably just acquire that in like a reference scan and apply it because it's very stable.
Right.
Especially the magnitude changes are pretty stable. It's just so like your RF pulse has, you get slightly different slice profiles across shots. That doesn't change so much with the zero or motion and things, unless the motion is really huge.
Okay. And my final question is the spatial resolution limit. Is it because of the SNR or is there any other limit that you have?
You say SNR?
Yes. Yeah.
Yeah. Right now, our SNR is brutal when we go to these high spatial resolutions. So I think that is the main one. We could just keep on increasing the number of shots so as to get larger and larger matrix sizes. This would be one strategy we could use, and so far we haven't run into a barrier there. But of course, as we do that, this is also going to increase or push out our volume repetition times. We're going to have poor temporal resolution, also poor spatial coverage, it's a trade-off of all these factors. So from an acquisition standpoint, we can still go at a higher spatial resolution, but SNR is really becoming the limiting factor. And then, how cooperative are your subjects, and will they come back day after day so that you can get decent SNR?
Okay, well, thank you very much. It was a great talk. Thank you.
I have a small question. That was a great talk. I'm just wondering, did you, this question is more like clinical class / physiological. Did you maybe try to look at the conditions that are affecting or might affect blood viscosity? Of course, like sickle cell is one, but it's kind of very rare. Like looking at the effects of endocrine pancreas, like insulin or glucagon on cells, like conditions that are kind of physiologically would result in the effect of blood viscosity. Did you move to pathology already or not?
No, so I've never done any clinical study. It's all been on healthy volunteers for kind of basic fMRI neuroscience applications. But I really want to get into clinical studies. So if there's potential collaborators here, I'd love to start getting into more clinical work, which I'm sure there are potential collaborators. You guys are a great bunch. But so I haven't looked at any particular, obviously now I haven't looked at any particular clinical pathology that impacts viscosity, but we have actually a lot of interest in viscosity, even for just basic neuroscience, understanding the blood signal alone, because the viscosity has a big impact on which blood vessels the red blood cells are going to go into, as you get down to to the microvasculature. And so for those reasons alone, actually, and we have now a new member of our lab, a new postdoc, who really specializes on understanding how hematocrit divides across the cortex from kind of a lot of computational work that he's been performing. And so we have some motivations from just the basic neuroscience aspect as well, which I think, these clinical models are probably much easier to work with in the sense that you're going to see larger variation in viscosity as compared to a typical healthy control. And also on top of that too, sorry, just one more aspect too. I guess I've also, in kind of previous work I've done, I've also looked at the impact then of red blood cells size and how that does impact then our relaxation times. And so we can also start to try to sensitize our MR signal to different red blood cell forms to some extent, which isn't directly related to viscosity, but viscosity would be a downstream effect of the type of red blood cell present.
Because one of my thoughts, this is really jumping far ahead in the future, erythropoietin has been kind of given to prematurely born infants to kind of decrease the injury of the brain. I'm oversimplifying when I say that. So I'm just thinking since it does affect the blood viscosity, that would be kind of super interesting to kind of dig in, because it's linked with therapeutics and also neuroscience, but also physiology. But that's just jumping far ahead. It's not fetus, but prematurely born infants.
Yeah, so I mentioned at the very beginning too this interesting finding now of red blood cell velocity is a function of kind of PO2. Not because this is something that like today we're looking into with, say, fMRI or with some other MR-based technique. But really, this is just like a new finding for me. It kind of blew my mind. And I think, yeah, if people are interested in somehow pursuing this further with non-invasive techniques, you know, because they were using 2-photon microscopy. So people want to look into this further with non-invasive techniques and really see how this then propagates downstream into humans. I think, yeah, there'd be, you know, this one study could spurn potential lot of interesting applications.
I agree.
So I'm glad that you were excited about it then.
Well, we've come up on the hour. Avery, thank you so much on behalf of the FNNDSC for giving this talk, and really appreciate you giving us some things to think about, taking your time today. So, appreciate it.
Yeah, thanks so much for the invitation, Jeff and Ellen and Gauravi. Yeah, it's really nice to meet you a bunch, and hopefully we can connect again sometime soon.
Sounds good. Okay, take care, everyone.